The overall goal of this procedure is to simulate central hypovolemia associated with hemorrhage in humans using lower body negative pressure and to assess the individual's capacity to compensate during this challenge.
Lower body negative pressure is a valid model of human hemorrhage which has been used to identify early predictors of hemorrhagic shock.
The main advantages of this procedure are that all measurements are acquired by non-invasive methods and that tolerance to hemorrhage can be safely assessed in the subjects.
Using lower body negative pressure, we developed the measurement of compensatory reserve, which can provide an early assessment of the progression toward shock during hemorrhage and is more accurate than the current tools to monitor vital signs. Compensatory reserve reflects the integration of all physiological compensatory mechanisms involved in the response to hemorrhage in an individual patient and therefore tracks the progression to shock with a high degree of specificity.
Prior to the study visit, instruct the subject to avoid caffeine, alcohol, and strenuous exercise 24 hours prior to testing, and to avoid eating at least 2 hours prior in the event that hemodynamic decompensation induces nausea. On the study day, begin by turning on all the equipment that requires warm-up and calibration: a data acquisition system, 2 infrared finger sensors, a capnograph, and a finger pulse oximeter. Synchronize all of the instruments with internal clocks by adjusting the timestamp on each instrument to match a lab master clock that should be used to mark time during the experiment. Next, escort the subject